Hello everyone, I am Nalini, Maharani Science College for Women, Bengaluru, Department of Botany. In this session, I shall be telling about the inhibition and regulation of enzyme activity. We all know the active sites of the enzymes are sometimes occupied by some inhibitors in place of the substrates. Those inhibitors are of two types, majorly the competitive inhibitors and non-competitive inhibitors. Competitive inhibitors are those which are going to compete for the active site since they are structurally similar to that of substrate molecules. While the non-competitive inhibitors are not going to attack the active site, but other than the active site, they will come and act upon the enzyme and change the conformation of the active site, so that makes the active site not suitable to fit in the actual specific substrate for that enzyme. Likewise, inhibitors are going to decrease the activity of the enzymes. By binding to enzymes to the active sites, they are going to reduce the compatibility of substrate and enzyme that leads to the inhibition of the enzyme substrate complex formation, and thus the products are not obtained. Totally, the reactions will be stopped. Preventing the catalysis of the reactions and also decreasing them up to the level of zero, the amount of the product produced by the reaction will be nil. Thus, the concentration of the enzyme inhibitors is going to retard the rate of the reactions. As I already told, there are two types of inhibitors, competitive and non-competitive inhibitors. They block to the active site in case of competition, or they attack the other side of the enzyme and change the structure of the active site in non-competitive inhibition. Majorly, they are also used as the substances which are going to kill the pathogen and correct the metabolic imbalance. So most of the times the enzyme inhibitors are nothing but the drugs which are going to kill the activity of the pathogen. They are also used in pesticides. Not all molecules that bind to the enzymes are inhibitors. Enzyme activators also bind to the active site of the enzyme, and they will help in the catalytic improvement of the reaction, enhances the catalytic activity. So most of the enzyme inhibiting agents are cimetidine, erythromycin, ciprofloxacin, and also isoniazid. These are some of the antibiotics which are going to act upon the pathogen, and they are going to kill the activity of the pathogen. So even the insecticides are all inhibitors. The presence of the inhibitors in the reaction mixture inhibits the activity of the enzyme, particularly the enzyme activity is totally or partially is reduced or stopped by the inhibiting agent. Coming to the first type of inhibition, that is competitive inhibition by competitive inhibitors. Such inhibitors have structural similarity with that of the substrate molecules, so they compete for the active site. If competitive inhibitor preoccupies the active site, then substrates fail to get that active site. But inhibition of this kind is always reversible. By removing the competitive inhibitors, the enzyme will restore its activity and convert into products. Example, the melanoic acid, which is going to have the same conformation with that of the succinic dehydrogenase, the active site of the succinic dehydrogenase, which has to fit in to the substrate succinate, but before succinate, if malanoic acid comes and binds to that particular active site, since it is structurally similar to the succinate, the substrate molecule, then the enzyme inhibitor complex frames in place of enzyme substrate complex, and no product is obtained. Okay, this is a very important example for the competitive inhibition type. Next, coming to the non-competitive inhibition. These are usually poisons. They never structurally resemble the substrate, but they are totally going to attack the enzyme molecule other than the active site, and they will totally change the conformation or structure of the active site and make it very much not suitable for the substrate conformation or substrate structure. Substrate structure is different, and you can see here the enzyme earlier it was having a round active site like this, but after the attack of this inhibitor, it has changed the structure of the active site and made it very much not suitable for the substrate molecule to come and bind. This type of inhibition is called as non-competitive inhibition, and it is very importantly irreversible reaction because it totally make the enzyme very much permanent damage will be caused to the enzyme, and it makes the enzyme very much inactive, and the activity cannot be restored. So it is an irreversible reaction. An inhibitor that binds to the secondary site of the enzyme other than the active site, this changes the shape of the active site and prevents the substrate from binding. This type of inhibition is usually done with so many poisons. We can see here diagrammatic representation of both the types, competitive inhibition and non-competitive inhibition. See, the substrate and the inhibitor molecules are structurally resembling. Here they are not structurally resembling. So that inhibitor molecule attaches to the enzyme molecule other than the substrate molecule, but this particular shape of the active site V-shaped here, but it becomes totally changed when the inhibitor is going to act upon it, and thus no reactions and no product formation takes place in both the type of inhibitions. We can see one more example here, how exactly competitive inhibitor structurally, the structure wise the inhibitor resembles that of the enzyme, but here it attaches to other site than the active site and makes the conformation of the active site different. So it is not suitable for this substrate to come and bind. And coming to the regulation of enzyme activity, we saw the inhibition of enzyme activity with competitive and non-competitive inhibitors. Now we will move on to the regulation of enzyme activity. How the enzyme activity is regulated within the body. Allosteric inhibition or feedback inhibition. Allosteric, nothing but the Greek word allo means other, stereos means space or site. Other site, allosteric inhibition is nothing but here the substance, usually the final product of the reaction, is going to act as the inhibiting agent, or it is going to attach to the surface, other surface of the enzyme, so that it is going to get changed. The enzyme is completely changed, the conformation, active site conformation is changed, and thus the inhibition of the substrate molecule to come and bind to the enzyme takes place. So here only difference here is allosteric other sites of the enzyme will be attacked by the final product of the sequential biochemical reactions on which the enzyme is acting. So usually the first enzyme of the series of biochemical reactions, for example in the formation of a particular amino acid, then that end product may cause inhibition the activity of the first enzyme in the sequence. That is called allosteric inhibition. Allosteric inhibition is always reversible, because since the product is present, that inhibition has taken place. But if that end product is completely consumed in the body, then there is no such end product to inhibit the first enzyme of the sequence. So when it is completely exhausted, then once again the first enzyme of the sequence will get activated, and it will proceed with the once again the formation of the same product. And likewise, very familiar, famous example for this allosteric inhibition is from the enzyme Escherichia, bacterial enzyme of Escherichia coli, which catalyzes the conversion of L-threonine into L-isoleucine, involving 5 different enzymes in this sequence. You can see first one is a threonine dehydrase, second one is acetolactate synthetase, keto acid, third one keto acid reductoisomerase, and fourth one is dihydroxy acid dehydrase, fifth one is transaminase. These enzymes which are operating in the formation of the isoleucine. So isoleucine is the final end product that we see in this particular reaction of conversion of L-threonine to isoleucine. Now we can see the schematic representation of this complete biochemical sequence. First, the initial substrate is L-threonine, which undergoes the enzymatic reaction with the threonine deaminase. So threonine deaminase is the enzyme which is going to give the enzyme substrate complex, the intermediate product. That is first one, that's why intermediate A. And then it acts upon the second enzyme, that is acetolactate synthase. And then intermediate B will be obtained, which is going to undergo the enzymatic reaction with third enzyme, keto acid reductoisomerase. And again fourth enzyme, that is dihydroxy dehydratase. And lastly, the fifth enzyme, that is transaminase, will convert the intermediate product to isoleucine. With this particular final product, that is isoleucine, is going to act as a feedback. Since it is the final product of the reaction, it is going to come and bind to the active site of the first enzyme, that is threonine dehydrase. The threonine dehydrase will get blocked, so that it makes the active site very much not suitable for the L-threonine to undertake the enzymatic reaction. So this particular isoleucine is going to block the enzyme threonine dehydrase, wherein it is going to change the active site conformation, and thus the conversion of the threonine to the intermediate A is completely stopped. And thus this feedback inhibition will be on till the isoleucine is completely utilized in this stream. Once again, if there is need for the isoleucine, then again the isoleucine is completely utilized. Thus there is no isoleucine to act as a feedback inhibitor. So once again the enzyme restores its activities. It is a reversible reaction, and again the L309 will come and bind to the enzyme 309 deaminase, and the process sequence will continue to give the isoleucine once again. So this is how the enzyme regulation takes place. Enzyme regulation is studied in Escherichia coli. In this sequence, only the first enzyme, that is 309 dehydratase, is initiated as a inhibiting agent for the by the isoleucine. Inhibiting agent here, it is getting inhibited by the end product, that is isoleucine. And then the activity of the enzyme is neither inhibited by any other intermediate complexes, enzyme substrate complexes, but it is only inhibited by the final end product. Hence we call it as feedback inhibition. Inhibition of the first enzyme, that is 309 dehydratase, is always reversible. When the concentration of the isoleucine in the cells increases, the activity of the enzyme is decreased, so that the production of isoleucine falls or stops. But when isoleucine concentration decreases, the activity of the 309 dehydratase will be restored, and it increases once again for the formation of isoleucine in the cells. So this is how the feedback inhibition works. And we can see the diagrammatic representation. Allosteric, once again, here allosteric means the site other than the active site. You can see allosteric site here is the this particular site, but active site is here. We can see the active site, initial active site will be almost the W groove is there, but here the active site will get distorted and it becomes only U-shaped, so that the substrate, and that is L309, fails to come and bind to the active site. Thus that enzyme is blocked, and we can see the distorted non-functional active site, and allosteric inhibitor will be looking here. We can call it as allosteric activator because it is going to tell the first enzyme of the sequence just to stop the reaction. It is not going to make enzyme non-functional as in case of non-competitive inhibitors like poisons. So we can call it as allosteric activator. Active site once again becomes functional when it is going to lose its activity. What is that? The final end product, when it completely vanishes, completely utilizes in the body, then it is going to convert as the allosteric activator, and once again tell the enzyme, once again makes the enzyme functional by making the active site, once again W groove is obtained, and into that W groove the L309 once again blocks, and the enzyme intermediate product will be on. So we can see here red colored they have shown for the inhibition action, and once the isoleucine is completely consumed, then it becomes green color, so we can also call it as activator now. Thus the regulation of the enzyme action takes place. Whenever there is need, the enzyme will be restored its activity. Whenever there is no need, the final product itself is going to regulate the enzyme action and tells the enzyme to stop the reaction for the time being, since there is no need of that particular end product. So this is the feedback regulation. To summarize the topic, in this session we discussed on the inhibiting agents, which are of two types, competitive and non-competitive. Competitive inhibitors are those wherein they are structurally similar to that of the substrate molecule, hence they compete for the active site. Non-competitive inhibitors, they completely damages the enzyme moiety because they are not going to attack the active site, but other than the active site they will enter the enzyme and they will deformate it so that the active site is not suitable for the substrate. And we also discussed today how exactly enzymes get their activity regulated by the end product of the sequential biochemical reactions. End product is going to come and block to the site other than the active site, and once again it deforms the conformation of the active site so that the substrate cannot make the correct enzyme substrate complex. It cannot bind it to the active site, and thus the enzyme activity is regulated or stopped. Once the end product is completely utilized, and once again if there is demand for that product, then the same thing is going to act as allosteric activator to once again make the active site conformation correct. So it is corrected, and once again the substrate comes and binds to it, and the sequence of biochemical reactions, for example in the synthesis of some amino acids, will be restored, and that amino acid is produced. And we saw the example of conversion of the L309 to L-isoleucine in Escherichia coli. This completes this session. Hope all of you have understood. These are some of the references. Please go through these textbooks and also the websites for better understanding. Happy learning. Bye. Good luck everybody.